Chapter 17 is important, and as I mentioned, 30% of your test, the final will be from this chapter. Okay, so what you need to know from this chapter. First of all, we are talking about two important features in cell biology and biology generally, and we call those the transcription and translation, and those are the central dogma in molecular biology. And central dogma state that the flow of information from DNA from genotype to a phenotype to make proteins. This flow of information, it should go through an intermediator, and that intermediator here is the messenger RNA. So information is flowing from DNA to RNA and from RNA to protein. We call this the central dogma of molecular biology. The process where DNA is converted or transcribed to messenger or give messenger RNA, we call it transcription. And the process where messenger RNA is translated to protein, we call it translation. There are very important elements associated. So DNA cannot go to messenger RNA without a very critical enzyme, we call it RNA polymerase. RNA polymerase is the enzyme that is responsible for making substrate, using a substrate and making a product. DNA is a substrate, and the product here is going to be a messenger RNA. What RNA polymerase do basically is transcribe, is copying. It's not degrading the DNA and converting it into RNA. No, it's copying the nucleotides the way DNA polymerase do exactly. But RNA polymerase making RNA, so there is no thymine. It will be any thymine will be replaced with uracil. In translation, this is where messenger RNA is translated. The key elements are going to be ribosomes and the transfer RNA. Those are the macromolecules that carry the amino acids during the process of protein formation. This is very important, information for this. And you need to know the difference between transcription in prokaryotes and transcription in eukaryotes. You know that the main difference between prokaryotes and eukaryotes, prokaryotic, they don't have nucleus. So their genetic material is scattered in the cytoplasm, okay? In a region, we call it nucleoid. And so both transcription and translation can occur together because there is no barrier. As soon as the messenger RNA is made, a ribosome can bind and start translation. In eukaryotes, it is different because they have nucleus. So DNA transcription is happening in the nucleus. And as soon as the messenger RNA is formed in the nucleus, it needs to be transported to the cytoplasm through nuclear envelope. There are some protein or some pores in the nuclear envelope where the messenger RNA can move from the nucleus to the cytoplasm where translation is going to happen. So it's two separate processes in eukaryotes. The other thing is in eukaryotes, RNA that is made need to be processed. It is not going to be ready for translation unless it is processed. And this processing involves three major steps. One of them is adding a modified guanine at the 5' end of the messenger RNA, and we call this a cap. And this is where the ribosome recognize and start bind and start initiate the translation. Also, there is a poly A is added at the end of the messenger RNA for stabilization. And also there are regions in the messenger RNA, in the pre-RNA that need to be removed. We call those introns. They need to be removed, excised out. We call it splicing, and exons only remain, and that will be the mature RNA, messenger RNA. So you need to know the difference in prokaryote and eukaryotes. You need to know that ribosomes in prokaryotes is different from eukaryotes, and this is why we can use some antibiotics to kill bacteria by inactivating the ribosomes without affecting our ribosomes, because structurally they are different. This is also, you need to know this. You need to know what is a transfer RNA is made of. So an RNA molecule single-stranded, but in nature, it is not going to be completely like one strand flying or floating. It made lots of secondary structure, so it will be very compacted, as you can see, for protection. Okay, the transfer RNA, its main job is like the Uber or Lyft that carry the amino acid to the polypeptide extension site. So this is, you know, the carrier for the amino acids. Where it carries exactly, it carries in a region at the 3' end of the transfer RNA. And we call this the amino acid accepting end. And this is at the 3' end of the transfer RNA. What else is unique about the transfer RNA? A region, we call it the anticodon. And an anticodon is opposite to the codon. So on the messenger RNA, there is a codon which specify amino acid, which is made of 3 nucleotides and specify amino acids. And on the transfer RNA, there is the opposite or the complementary of this codon. We call it anticodon, where this is a docking is happening between the transfer RNA and the messenger RNA, and then the ribosome will going to make the complex to start the process. So again, codon on the messenger RNA, anticodon on the transfer RNA. You need to know this. Each amino acid is specified by 3 nucleotides. Okay. If I give you a piece of DNA and ask you to do translation, do you know how to do it?
We had to do it for our homework, but you know, sometimes we would get like T's. Are we right to transfer the T's to a U? So if it was like TGA, are we right to say it would transfer to UCA?
Okay, let me write down here. Say if you have a piece, say, let me do something very quick. Say I have a piece of DNA here. Let me do this.
Because I think in our homework we had like just a long code, so it was like GCATTUAC whatever, and
Okay,
we
let me
had to
write.
like
Okay,
transfer it out in threes.
GCATTACCGGCATTAG. Say this is a piece of DNA here. Okay, and this piece of DNA is going from 5' end to 3' end. Okay, and the complementary is going to be 3, and then this is G, so it will be C, G, and then it's T, and then A, T, and then 1, 2, 3, 1, 2, G, T, and then A, T, C, and that will be 5. Okay, say this is a piece of DNA here, and I said, okay, which one of those I need to do transcription first. So which one of those will be transcribed? You have a piece of DNA, and you have 2 DNA like complementary, and say you have like in this area you have a promoter. So say you have a promoter sitting here, promoter. Okay, this is a promoter, and let me write down P stands for promoter. Okay, so now one of the strand we call it the coding strand, one of the strand we call it the template strand. So So which will be the template strand? Which will be the coding strand?
Is this got to do with like leading and lagging? So would the
No,
top one
no,
be?
no. You have no correlation.
Oh.
You are talking about DNA replication and DNA transcription. No, there is no connection at all. The enzyme involved in this process is an RNA polymerase. It's not a DNA polymerase. So it is not basically replicating, it is transcribing it, making copy, and when it do this kind of copying, every time it will meet an A, it will put the complementary as a U. Okay, because this is an RNA molecule. But now my question, which strand of those is going to be the template? Which strand is going to be the coding? So if I give you this piece of DNA, I said, go ahead and do transcription and translation. Which strand you're going to select to do this process?
The top one for coding and the bottom for template.
Yes. So the one which is going from 3' to 5' and we call this the template. Why is the template? Because as soon as the messenger RNA is going to be formed, it will be anti-parallel. So the messenger RNA going from 5' to 3'. So we can't use that one to make a messenger RNA from 5' to 3'. So it will be parallel. In nature, this is not happening. So in nature, it is the opposite. It is anti-parallel. So since messenger RNA is made from 5' to 3', so the strand that is going to be used for the template by the RNA polymerase is going to be the one which is going from 3' to 5'. This is very, very important, okay? So now say I need to do transcription, sorry. So the messenger RNA is going to be from 5' to 3'. So it will be complementary to this, right? Since it is complementary to this, so it will be exactly like that. This is why we call it the coding strand. The only difference will be every single T, you are going to replace it with U. Okay? So now you have your messenger RNA. So your messenger RNA is basically the same like your coding strand, with one difference, which is there is no T in the messenger RNA, so it's going to be replaced with a U. Okay? So now you have your messenger RNA. In prokaryotes, you don't need to do anything. It will be ready to go. In eukaryotes, it needs to be modified. So if there is some introns that need to be removed, a cap that need to be added, a polyadenylation site that will be added to the 3' end, this is in eukaryotes. So for the sake of simplicity, now how this is going to be translated. In prokaryotes, there is a region, we call it ribosomal, we call it Chine-Delgarno sequence, or ribosomal binding site. This is where ribosome binds. We call it Chine-Delgarno. It's like maybe 5 to 6 nucleotides in length, and it is very rich in purines, meaning guanine and adenine. Most of the bacterial genes, they have this Chine-Delgarno sequence where ribosomes sit and start the process of translation, okay? If I will give you, I will identify the Chine-Delgarno and say this is the Chine-Delgarno here, so you are going to start with the first AUG. If I didn't specify, then you go ahead, complete the process of translation. How you do this? You take 3 nucleotides at a time. GCA, there is a wheel and there is a table that you can use. The wheel is easy. The wheel, as you can see, made of 3 circles, okay? And when you read from inside out. So if I'm reading GCA, you take each nucleotide from each wheel. So the first wheel, I will take a G, which is here. The second wheel, I'm going to take a C. So now which one? It will be this one, right? I have, this is the only option I have. And then from the third wheel, I will take the A. So I need to go G, C, A, okay? And that will be alanine. That will be alanine. So I put alanine here. Then the second codon, U, U, A. U, U, okay, it is U, first wheel, U, the second wheel, and U, sorry, A, the third wheel. U, U, A, that's a leucine. So I put leucine. And then I go to the 3 C's. First wheel, C, and then the second wheel is going to be, this is a C, and then the third wheel is a C. So C, C, C is a proline. So I put proline, and so on. If I need to, if I use the table, the same. The table is made of those like one horizontal and two vertical line. We call it 1, 2, 3. 1, 2, 3. So for example, if I go G, G, C, so I go to take the G from the first, okay? This is a G, and then another G, so I go here for the other G, which is here. So this is a G, this is a G, and then the third nucleotide will be a C. So G, G, C, it will be here. G, G, C. G, G, and then which one I need to select from those, the one which will go the intersection between the two together. So this one and this one, they will intersect in this area. So G, G, and then C, that will be a glycine. So I'll select glycine and put glycine here, okay? And then AUU, for example, AUU. So again, A, and then a second A, and then those intersect together, A and A in this area. So A, so it's AUU, sorry, not AAU. So it's A and then U, A, U, U. So A, U, U will be here. This is an isoleucine. So I put isoleucine. We call those the 3-letter code. 3-letter code for amino acid, as you can see. So alanine, it's ALA. Arginine, ARG. Aspartic acid, ASP, and so on. And there is also something called the 1-letter code. So the 1-letter code, the amino acid is represented with 1 letter only. So alanine is A, okay? Arginine is not going to be an A. Arginine is going to be an R. And aspartic acid is a D, and so on. So if I give you a piece of DNA and said, go ahead and do transcription, so you need to create the messenger RNA, the most tricky part, you need to select which strand you are going to select for transcription. As I mentioned, again, you need to select strand which is going from 3' to 5' as your template, and make the messenger RNA from 5 to 3. And then when I ask you to do translation, 3-letter code, each codon will give an amino acid, you put the 3 letters. And if I ask you to have 1-letter code, you need to convert 3-letter to 1 letter. You are going to see this in your test for sure. Yes.
If you asked for the 3 to 1, would you give us that chart?
Definitely, yes.
Okay.
That will be, I need to give you this in your, yeah. You have to have this. You don't need to memorize any of these.
Okay.
You know, no memorization for anything. You are going to have those, the table and the wheel, so you can use those to do the translation, yes.
Okay, thank you.
Yeah. Any question for me?
So for chapter 17, we basically have to know the flow of DNA, how it's transcribed, how it's translated, and you want us to know the difference between prokaryotes and eukaryotes.
Yes, it is important. Yes. Also, you need to know so two things. Yeah, you're right. So we need to know the difference between prokaryotes and eukaryotes in the process, so that the difference between the for transcription in prokaryotes and transcription in eukaryotes, how they are different. Maybe the difference between the prokaryote ribosomes and eukaryote ribosomes also. And the most important thing would be how to do transcription and translation for a piece of DNA. If I give you a piece of DNA, I ask you to do that.
Sorry, I'm just writing that down.
Yeah.
I'm pretty confident for, I'll just look at the email 1 second. I'm pretty confident with chapter 16, 14, 10, and 9. What should I be brushing up on for chapter 5 and chapter 7?
Chapter 5 is very important, macromolecules. So you need to know difference between monomers and polymers, the building blocks for proteins, for fatty acids, polysaccharides, all this good stuff. And you need to be able to also to, if I give you a structure, you need to be able to tell me if it is a sugar, if it is a fatty acid, if it's a protein, this kind of, amino acid. You need to know the difference between those. I'm not going to ask you like to memorize structures, but this is the basics for chapter 5, macromolecules. Those 4 classes or 4 categories of macromolecules, you need to be familiarize yourself with those. Chapter 7 is the membrane, basically a little bit about the structure of the membrane, and again the hypotonic, hypertonic things, osmosis, water flow from where to where, ions flow from where to where. Very little, not much. Not much.
Okay. I think I'm pretty confident with the rest. I'm going to brush up on 17, 5, and 7 tomorrow, but I
17
think,
is, 17 is 30% of your test will be 17. So that's a good idea to focus on this 30%. And then, you was tested for the other. So at least the information is still kind of fresh in your mind. So,
yeah.
you need just to go through, read it quickly and just to refresh your memory. 17 is going to be kind of new information for you. So yeah, you need probably to put more time on this.
Okay. Thank you very much for your time.
No problem, no problem. What is your name again?
Millie.
Millie. Okay, thank you, Millie. Where are you from, Millie?
England.
Oh, yeah. Very clear. So you there already in England, or you are here?
I'm in England at the minute. I was supposed to come back to start school, but obviously the travel ban and coronavirus hasn't gone to plan.
I know, yeah. It's affecting everybody, I know for sure. Okay, Millie, I really enjoy having you in my meeting. Thank you very much and good luck. And just email me if you have any further question, anything that was not covered, email me about that particular subject, okay?
Okay, yes, sir. Thank you very much.
Thank you much. You have a great day, okay? Bye-bye.
You too. Bye-bye.
Bye-bye.